Hi, I'm Angela Hornsby. I'm the curator of the Philip L. Wright Zoological Museum at the University of Montana. I have with me today, this is Sam Getty.
Hi, my name is Sam Getty. I am studying wildlife biology here at the University of Montana, and I am a senior this year.
The Phil Wright Museum was founded in 1897. We're almost as old as the university itself. And our oldest specimens are actually even older than that. They're from the 1880s. So we have a lot of cool stuff that's really old and serves as a good baseline for comparison when you're looking at more recent things versus old things and all the changes that have happened in between then. So the Phil Wright Museum is a research museum. If you're not familiar with that, it's more of like a library than a museum that you would visit to look at exhibits in. But instead of books, we have specimens. And we have those specimens for three major reasons. The first is that so students can learn what different species look like. The second is that so researchers can learn what different species look like. And in fact, researchers use the collections to determine whether they have found a new species, to describe that species, to tell us how we can differentiate it from other species. And the third major use is for understanding how changes in the environment affect the birds and mammals, which is what our museum focuses on. Today we're here in the prep lab and we're going to talk about the life of a specimen. So what happens between the time that we get a specimen and it goes into our permanent research collection. When someone donates a specimen for us to prepare, it goes into our freezer first, and once we're ready to prepare it, we'll take it out and thaw it. So here's actually a horned lark straight from the freezer that we haven't prepared yet, but it's next on the list. So once the bird is thawed and ready to be prepared, we will choose a data card. This is the bird data card since we're looking at horned larks today. And this is the place where we fill out all the information we have about that specimen. What species it is, where it was collected, when it was collected, who collected it. We take a whole bunch of measurements, like we'll measure the length of the tail and the length of the wing, different measurements of the bill, and we'll do some molt notes and other notes based on the external condition. And once that's all done and on the data card, then we'll actually start to skin it up.
Alrighty, so once it's all thawed and we have all the measurements, we start off by cutting the bird open. Then once we are done with that, we will try to get the body out and get ready to stuff the bird. So this is the body. Once we get it out, we'll hand this over to Angela in a little bit. Then we will start to stuff the skin. So I've already cleaned it out, and next we will take a wooden dowel and wrap it in cotton to make a little body. And then we will just insert that into the skin. And then the next step would be to close it up and sew it closed. Alrighty, so once a skin is stuffed, we will take it and we will put it onto a pin board like this. And it'll stay like this for a week or two. And after that, we will pull it off of the pin board and Angela will take it and get ready to put it into the collection.
So one of the important things that we do with the leftover body, once it's out of the skin, is to take tissue samples. And we'll normally take tissue samples from multiple organs. So the heart, the liver, the lungs, the muscle, and for mammals also the spleen. And that gives researchers a lot of different resources if they're wanting to look at RNA or protein that are found in different organs, or if they just want the DNA that would be found in any organ or any tissue. And we put it into our -80 freezer. So this is our special freezer that is kept at -80 Celsius. And that means it is extra cold to help preserve all the tissues for as long as we possibly can. The idea here is to preserve things that future researchers might want to use, whether that's 10 years from now, 100 years from now, or 200 or 300 years from now.
So once Angela is done taking the tissues, she gives us back the body cavity, and we will go through and clean off as much of the flesh as we can. We'll also take out the internal organs, and when we do that, we will often go through the stomach contents and the contents of the crop if it's a bird, and then we will note those on the prep card. This way researchers can find out what the bird was eating, perhaps where it was from, and various other things that you wouldn't really be able to see.
So once those skeletons are dried down, we feed them to our beetle colony. And basically all the larvae, and the adults to a lesser extent, will go through and eat all the extra tissue off bones. So this is a set of deer bones that we have that are broken open, so that you can actually see them inside eating all the marrow from inside of the bone. Once the skeleton is cleaned off, we will take it out of the bug colony, clean all the bugs off as best we can, and then freeze it just to make sure that there aren't any eggs that will hatch out of it still.
So this is what it looks like when it comes out of the bug colony and out of the freezer. So we'll take all this and we will soak it in some nice hot water to try to clean everything off of it. And once we do this, we will get a nice, clean, finished skeleton like this, which is all ready to go and be labeled and put into boxes to go into our collection.
The last part of the preparation process is to assign a catalog number to the bird. For instance, this is a horned lark that has been assigned the number 22111, and that'll be its permanent number for all of time. And we write 22111 on the tag, and then we also write it on every bone that we can. Now it's ready to be installed into our research collections. So now we're in our research collections room. This is where we keep the vast majority of our 22,000 specimens that are used for research purposes primarily. We have a series of compactors in this room, and basically each bank of cabinets can move down and make a new walkway as needed. And the compactors are really nice because it helps us fit about twice as much into this room as we'd normally be able to. So whenever we have a specimen that's ready to go into the research collections, we find the drawer that it belongs in with all of the other specimens from that same species, and we put it right there in order. And now our specimen that we just put in here is right next to some specimens from, this one is from 1900, this one's from 1927, this one is from 1892. So all sorts of specimens. These are all just horned larks that will help us understand how horned larks change over time. So in terms of keeping track of all the data for the specimens, this is how we used to do it, just on a paper ledger that has the specimen name and when it was collected, who collected it, and where. And nowadays, instead of just keeping things in a paper catalog, we put all of our data online, and it's open and freely available to anybody across the world who wants to look at it. We use this collection service called Arctos, and from Arctos you can access all of our records. For instance, here's the record for that bird that we just installed into the collection. There's another website called GBIF where our data eventually go, and GBIF aggregates data from across websites, whether it's museum collections or observations or fossils or other records of that species. So you can see where all of our information on horned larks come from. Where does all this data go to? Researchers use it for all sorts of different things, but you use it also if you've ever used a field guide. So for instance, this is a horned lark page. We have the length, the wingspan, the weight. All those are measurements that were taken and aggregated from museum collections. And we also, for horned larks, since it's a wide-ranging species, we have information on what different horned larks look like in different areas. And this is all based on museum specimens as well. People have lined specimens from all different places up, looked for the differences, and recorded that so that it's now a guide for anyone who's interested in horned larks. Thanks for joining us today on behalf of myself and Sam. We hope you learned a little bit about what the Natural History Museum does, and if you're ever on campus, I hope you come to visit us in person.